Hi there. Thank you for coming along to this talk, and thank you to the organizers for the very generous idea of asking me to give a talk. For the next 15 or so minutes, I'd like to talk you through some of the thinking currently in models of cancer progression, and in particular some of the ideas coming out of our group in Australia and in Oxford around invasion, plasticity, microenvironmental stress, and cancer progression. And in particular, the emergence of some thinking around a new model of cancer in which we think of it as an autonomy-seeking lesion, and something approximating a form of organized crime rather than something sinister. Here is a brief outline for the talk, and if you don't recognize the picture on the right, it's a painting of Ned Kelly, who was a very famous Australian outlaw and continues to be a divisive figure along with his gang, partially because it's not clear to what extent this kind of criminal activity in Australia at the time, and particularly from the Kelly gang, was the result of basically the absence of welfare and bad, corrupt policing, people simply trying to make ends meet, and how much of it was, in some sense, malignant. But what I'd like to start with is to convince you that wherever the truth may lie, we are in dire need of new myths and models for cancer, because we are doing very well in the fight against cancer, despite dramatic improvements in genetics and in other areas of endeavor. I'd like to tell you a little bit about the microenvironment in which the tumor exists and the kind of stresses that it's subjected to in that environment, which we now believe are basically inducing plasticity, epigenetic plasticity related changes in the cells. I'd like to tell you a little bit about the atavistic model of cancer, which tries to integrate some of these ideas, but I argue fails to explain many of the key aspects of cancer progression. And I hope to also show you some work coming out of our group around uni and multicellular programs, not only in cancer, but also within the human genome, and how we might think of autonomy seeking in that framework, which leads on to the idea of cancer as essentially a failure of cooperation, or a failure of support for cooperation amongst multicellular ensembles, and therefore of cancer as something like a form of organized crime. And if there's time, I will also show you some results of some mathematical models also coming out of our group. I think a good place to start is to think about the kind of words that we use around cancer, typified by books with titles like this. In the hospital, the words we use, particularly when patients are around and we maybe don't want them to hear something or understand everything, we say that something is sinister or it's a malignancy. When we talk about cancer in general, we use words like invasion, aggression, and those suggest, those words reveal a lot about our myth, in the traditional sense in the classical strict sense of that word, around cancer, that it's an evil force bent on destroying us, and that it's basically like a foreign invader. In fact, this is more or less how people used to think about crime in society. We used to think people were born into sin or possessed by the devil or whatever, and that's why crime happened. Of course, we now are aware that it's the absence of welfare. Basically, if people don't have food to eat and safety and education, then if there's gross inequalities, then crime turns up, and particularly organized crime. Whereas when those conditions are not in place, crime is relatively uncommon, and even where it occurs, it tends not to be as damaging to the society around it. So models of cancer progression obviously have been around as long as cancer research has been, and they aim to explain how we go from a basically normal piece of tissue to disseminated metastatic illness. The original models focused on accumulating random mutations, but it quickly became clear that that simply wouldn't explain a lot of the dynamics around cancer progression and invasion. We now understand that it's epigenetic changes that drive progression, but we don't really understand how. And at a high level, there are many challenges. I list 2 or 3 here, but it's not clear how cancer goes from a small cluster of basically not particularly malignant cells to being able to grow its own vasculature, evade the immune response, and so on. And also, we don't understand what is the role of the microenvironment. And of course, those questions have implications for what kind of treatment strategies make sense. The tumor microenvironment, very nicely illustrated here on the right. We don't understand it very well, but basically it's composed of the tumor cells, but also some normal cells, the extracellular matrix, and various immune and other cells. And this is basically the environment that the tumor is initially evolving in, and even at the late stages. Now, we have evidence increasingly that the tumor microenvironment is essentially a hostile environment for the cancer cell. The cell there is subjected to physiological stress, including nutrient deprivation and lack of oxygen. It's also subjected to attack from immune cells, and presumably all of this is happening because of crowding. So initially, a cell picks up a somatic mutation, which either interferes with apoptosis or confers on it a proliferative advantage of some type. And the resulting overcrowding is causing at least the initial physiological stress responses. So when we think about the epigenetic adaptations of tumor cells to this kind of stress, some of them are shown here at the bottom of the image. So there's increasing motility, which promotes escape from the microenvironment, and also cell-cell cooperation during that invasion process, leading to collective cell migration. The epigenetic changes caused by this stress, we argue, are basically those associated with cell plasticity. And plasticity is in some respects simply a mirror of cancer progression. And there are other changes apart from loss of stickiness and motility. There's resistance to apoptosis and some metabolic changes that help the cell to survive its hostile environment. The atavistic model turned up about a decade and change ago, and it's actually based on an old idea, which is that cancer in some sense represents a reversion to a unicellular state. And that's exactly what the atavistic model postulates. Some evidence in its favor here on the right, looking for instance at the bottom left-hand panel, we see that as the higher Gleason scores are associated with essentially older or unicellular genes being turned on, and that's recapitulated in the central panel, which shows where in the phylogenetic tree essentially the cancer mutations can be associated to. The atavistic model is interesting, and it's very ambitious. It resolves some long-standing problems, including how cancer goes from being just a cluster of cells to having these superpowers, including immune evasion that we were discussing a few minutes ago. And there is some evidence in its favor. There's also quite a few things that it doesn't explain. For instance, cancers are highly innovative. They learn how to grow their own vasculature. They learn how to evade the immune system, and those are not things associated with unicellularity. It also fails to explain the existence and the place of stem cells. In particular, even in studies that support the atavistic model, there's nowhere really to place the stem cells on the graphs. But perhaps to me, most strikingly, tumors are multicellular. the cells are cooperating with each other in very sophisticated ways, and that's actually a characteristic of cancer. So it's not totally clear how that would be explained by a simple reversion to unicellularity. Here's some work out of my group in Australia. So if we look at molecular programs in humans and classify their constituents as being either unicellular or multicellular. So blue is if there are more multicellular genes, and red is if there are more unicellular genes. We can then calculate we can essentially get a complex network of interactions between these programs, and the genes in these programs are grouped because the program is doing like something related. So, for instance, neurological development or the growth of vasculature or whatever. And then we can try to partition those networks by various means. For instance, as here, this has been done using modularity maximization. And at the top, you see the entire complex network of programs, and it has exactly two submodules shown here on the left and the right. The module on the right is entirely unicellular orientated processes like DNA repair, cell division, and respiration. And on the right, we have the newer programs like brain development, vascular development. And there is some interesting substructure underneath. But this is a surprising finding, at least to us, that you can taxonomize the human genome in this way. And we can look at that data from a different angle. We can plot on the x-axis the proportion of genes in each program that are unicellular versus multicellular, shown in red and blue respectively. And on the y-axis, we can plot how connected that program is to other programs. And we see that the older, more unicellular dominated programs are much more connected to other programs, including the multicellular ones. And the multicellular programs are, relatively speaking, less connected both to each other and also to the unicellular programs. And if we go further down, we see that actually that motif is recapitulated at the next level. So if we look at the unicellular submodule, its own constituent modules are, the more internally connected they are, the more ancient they are in some sense, and that is not true for the multicellular module. Its submodules do not show this relationship. So you can think of this as a sort of a city in which the ancient city center is very tightly connected, and then there's a newer perimeter, kind of like a donut outer ring of suburbia around the old center, which is less connected to itself and also less connected to the center than the center is to itself and to the suburbs. And all of this is suggesting, we argue, a new model of progression here, schematically illustrated. And if we start with complex animals at the bottom right, we see that an in situ tumor caused simply by a somatic mutation that causes a proliferative advantage and therefore causes stress due to hypoxia or whatever. Basically, the adaptations to that stress are going to be to do with increased motility to escape the local environment, loss of cell stickiness, and other changes that leads to autonomous cells, which would, in some sense, be unicellular. But then, in order to survive the next set of stresses that they are subjected to, they basically reform into simple animals that cooperate, in a colonial sense, with each other in order to evade immune responses or to share resources in some sense, and that essentially represents the growth of a sophisticated criminal organization, which in some respects is not evil. It's simply trying to. It's like one man's revolutionary is another man's terrorist, and arguably the tumor is simply trying to survive and to be left alone and to have its autonomy. And all of this suggests a unifying motif for progression, which is that it's actually not to do with unicellularity, and it's not to do with the acquisition of somatic mutations, and it's certainly not to do with malignancy or being evil or anything of that type. It's simply that local tumor microenvironmental stress leads to a failure of support for large-scale cooperativity, basically. If obeying the law is more likely to get you killed, then sooner rather than later, people stop obeying the law. And obviously that's a very high-level model, but let me show you a little bit of evidence in favor of something more quantitative. These are simulations from a PhD student of mine. So what we do is we put an agent-based model down the tumor cells, which initially are normal, have some simple genome. They can stick to other cells, they can move according to certain rules. And we let the system evolve. We then subject the microenvironment to stress, and we only allow a certain number of cells to survive. So beyond a certain, you start off with 1,000 cells, but you can never have more than, say, 1,200. So as cells proliferate, because of an in situ lesion, you get crowding, but we only allow a fixed number to survive. And then we look at the genetic, between inverted comma, profile of those cells. And on the left here, what you see is that as generations go on, at the beginning, there's not much change in cell stickiness because there's not much stress. But once stress reaches a certain level, the whole population is being selected for cells that are able to escape. And to escape, you need to delete coherence or reduce cell stickiness. So we see a drop. And on the right is how mobile a cell is. So initially, the cells are not particularly highly mobile, but the ones that are more mobile through epigenetic changes are more likely to escape the hostile environment and survive. And so as the generations go on, you see that the cells essentially become more motile, and there we have some other results around this unpublished work, but I hope you understand the kind of idea that we are trying to convey with this work. In terms of implications and thinking about future work, there are obvious diagnostic, prognostic, and staging applications, because the more autonomous, in between inverted commas, a lesion is found to be, presumably the more advanced that tumor is. But perhaps more importantly and more down the line, we argue that chemotherapy, immunotherapy, and radiotherapy basically promote progression. They harden the criminals. This is like the police coming to beat everybody up and arrest everybody without due process. Initially, those things have success in suppressing crime, those strategies, but in the long term, they basically guarantee the existence of sophisticated, increasingly violent criminal organizations, and ultimately a civil war. It's not hard to find evidence of what happens in human societies when those measures are applied. You can just turn on the TV. The implication is that we should perhaps think about fighting cancer the opposite way, the way we fight quite successfully crime. We've brought crime down to very, very low levels historically, and it hasn't been through policing, and it hasn't been through violent law enforcement. It's mostly been through welfare. So we give people safety, education, we give them jobs, and societies that have those conditions are very low in crime, and societies that don't have those conditions are very, very high in crime. And we have some early mathematical support for a model of this type. And lastly, I'd like to acknowledge my collaborators and my students, some of whom are at QUT, some of whom are at University of Queensland, and some in Oxford. I'm supported by a future fellowship of the Australian Research Council, and I'd very much like to thank Andy for thinking of having me give a talk at this conference. I'm sure I'll enjoy the rest of the conference. I very much look forward to listening and asking some questions. And I don't know if we have any time for questions for this talk, but if we do, I very much look forward to them. And thank you again for your attention.